Great. So like you said, I'm Zara from CATS. My email's at the bottom. Feel free to email me at the end of this PowerPoint if there's anything you have questions about in particular. And we will be today talking about text just appeared from there, vision, audition, and smell and taste. And we'll just spend the time kind of going through the processes of these systems. Just, I think what I found most important in second year was just kind of getting the order of all the different systems in your head, and then you can make more sense of the clinical applications of such and sort of the neuronal processing once you have the specific order of the systems. Cool. Okay, so we haven't got loads of time, so just 2 seconds to have a look at these images. Kind of start thinking about what's coming to mind. If anyone wants to yell anything out, they have to turn your camera on, but feel free to. Okay, so one of my images here is the retina. So this is where everything starts. Our cells in the retina are myelinated, and we have 5 layers in the retina. So we've got the ganglion cell layer in a plexiform, in a nuclear, outer plexiform, and outer nuclear. And then we have, of course, the photoreceptors beyond from that. And we have maximal acuity. So we've got our best point of vision at the fovea of the retina. And that's because all the inner cell layers are thin and they form a pit at the retina, and this pit has a very high density of cones, and the nerve body cells are shifted from this pit, so light has a really direct path to these cones. There are also no blood vessels here, so we just got this high acuity. And that's kind of important factually to know, but also what does that tell us kind of early on about the function of these photoreceptors and also the function of light, which is kind of interesting to think about if you're considering neuro for third year, how important light is in the system. Okay, so at the top, I've just popped the stuff we've already covered. Moving on to photoreceptors, we've got our photoreceptors here, we've got our rods and our cones. So photoreceptors contain retinal, and this is a photosensitive pigment, which is bound to a membrane protein called opsin. And rods and cones, as we can see here, are composed of inner regions. So we've got nucleus, mitochondria, other cellular apparatus, and then our outer segment has got these folds, and this gives us this large surface area in the cell membrane that will collect photons, which are obviously really important for visions. And we've got these rods have this larger outer segment than cones, so they've got a greater sensitivity in dim light, and that's not the only difference between them. I won't go through this whole thing, but I've got a big table here just in case in revision that this is useful. So you can see rods have more photopigment, cones have less photopigment. However, it is worth mentioning that cones have 3 types of pigment. They've got 3 types of photoopsin. So they've got a short wavelength activated, which responds to blue wavelengths, a middle wavelength which responds to green, and a longer one which responds to red wavelengths. And if you want sort of an interesting clinical point for essays, colour vision affects these pigments. So you can have horrible spellings, but due to anomality, which is the most common colour blindness, and this is an abnormal wavelength pigment of the middle green wavelength pigment, and anomaly is the abnormal red wavelength pigment, and these defects are all X-linked, which is why they're rare in females. Okay, so we've kind of covered a few of those differences now. So I think once you kind of rote learn these, you sort of get an understanding of the differences between the rods and the cones. So we will move on from that. Okay, so we've talked about our retina and we've talked about our photoreceptors right at the back here, our rods and cones. So now we'll move all the way down to our ganglion cells. And as we can see here, they output to the optic nerve. And these converge on the optic disc to form the optic nerve. And if you look at sort of ophthalmology images of the back of the eye, you can see this. You can see the optic nerve, and that's important for your exams to know how that looks. And again, this bundle of fibers is unmyelinated as they're intraretinal, and as they leave the retina, they then become myelinated. And we can divide our ganglion cells into magnocellular and parvocellular. So only 5% of these are magnocellular. These are large cells, they've got lots of myelin. We've got 100,000 of these per retina, and they produce transient bursts of action potentials in response to a sustained stimulus. They're also rapidly conductive with a large receptive field, and they're wavelength insensitive. Okay, so just think about how these differ now to our parvocellular cells, which take up 90% of the ganglion cells. These are smaller cells, they've got little myelin, and they sustain action potential firing in response to a sustained stimulus. However, this is slower conduction with a small receptor field, and these are typically wavelength sensitive. And I think there are a few ways of kind of remembering which one's round. I kind of remembered that the sustained action potential firing would be sort of the slower response. I think is probably a good way to think about it. Okay, so slightly moving on from that, one other thing to mention are, again, if you want sort of extra essay mark points, Muller cells, and these are vertically oriented glial cells, and they form a layer adjacent to their inner limiting membrane, and they maintain the ionic composition of extracellular fluid, and those are relevant to ganglion cells. Cool, okay, and then another sort of point of interest for essays, diabetic retinopathy. So the inner retina blood supply comes from the central retinal artery, which is a branch of the ophthalmic artery. And vessels run within the choroid of the retina, and when someone might have diabetes, hypoglycemia can cause pericyte death and thickening of the basement membrane, and this can lead to incompetent vascular walls. So therefore you can have permeable walls leading to macular edema, and this leads to blurred vision and blood vessel proliferation, which therefore then will also bleed, and this destroys and clouds the retina. So that's one to know about. And then another one being, just in a different colour, macular degeneration. So this causes blurred or no vision in the central visual field due to a damaged macula, and this can either be dry due to yellow cholesterol deposits, or it can be wet due to abnormal vessel growth, and this damages again our photoreceptors. So two kind of points that are good to know about. I'm just looking at my notes, by the way, sorry, that's fine. Okay, sort of the big clunky revision point is visual transduction, and this is effectively the absorption of a photon by a pigment molecule within a photoreceptor, which you can see here, and the cascade of events that follows. I will go over this very quickly if it is helpful for revision. So we see here we're in the dark. Our photoreceptor is transmitting a excitatory potential. It's depolarized, and you can see here in the light it's hyperpolarized and we haven't got any glutamate release. So therefore, what happens when the photoreceptor absorbs a photon? So rhodopsin is isomerized, and the 11 cis form of retinal that we have on the dark side becomes an all-trans form of retinal. And because rhodopsin is unable to bind to this conformation, it then detaches, and this intermediate configuration of meta-rhodopsin 2 activates transducin, which is this T, and this is a G protein that activates PDE, phosphodiesterase, that converts cGMP into 5-GMP. We now have less GMP. So if we now look at the second dropped down image, normally GMP is activating these channels on the photoreceptor, resulting in sodium influx. However, as we have less cGMP, these channels are shut, decreasing our depolarizing sodium current. So we hyperpolarize to -70 millivolts, calcium levels are reduced and less glutamate is released. So we have a IPSP. Okay, that was long, but hopefully that all makes sense. That's something I would definitely focus on revision. And yeah, this basically is a negative feedback loop that can be switched on and off. So it's really effective and again, really responsive to light. It's something I would think about for exams. Cool, okay, let's move on to think about retinal processing. So we really only talked about ganglionic cells and rods and cones, but we have loads of cells in the middle here. So cones synapse, as we can see on this diagram, onto horizontal cells, and these are GABAergic cells, which provide center around antagonism. What this basically means is these horizontal cells are inhibitory, they synapse back onto the cones surrounding them, and neighboring horizontal cells are connected to each other. So therefore each cone is inhibited in a network by the activity of neighboring cones. So this produces a center surround antagonism, and it characterizes all the receptive fields of the cells as the visual system develops. Okay, moving on to amacrine cells. In the red there, again, these are inhibitory and they contribute to inhibitory surrounds, basically by informing neighboring cells of what the other cells around them are doing, and they normally fire with GABA, glycine, or dopamine. So yeah, it's sort of a similar response. They contribute a bit to on-off responses and inhibitory surrounds. And then of course we have these bipolar cells, of which I did this extra diagram. So these respond to local contrast. So they have so we have on-center and off-center cells with different receptor fields and different glutamate receptors on dendrites. So our off-center cells respond to glutamate with AMPA current, leading to depolarization, and our on-center cells respond to glutamate by metabotropic glutamate receptor activation and hyperpolarization. So what bipolar cells are basically looking for is this idea of local contrast. So I guess if you were to look at a wall with a light on it, your eye would be focused on the shadow. So your eyes naturally look for local contrast. They look for the difference between two colors. Even on the wall behind me or on my face with the lamp on, your eyes create the shadows. So the receptive fields of bipolar cells can be turned on or turned off depending whether whether the stimulus is light in the center of the receptive field or whether it's darker in the center of the receptive field. However, important thing to note about this is neither bipolar cell responds to overall stimulation. So a bright light, simply looking at a very bright light up and close, wouldn't give as good, I couldn't say it couldn't give any, but it wouldn't give as good stimulation as looking at a shadow on the wall. And it's like I said, it's the receptors that give the on-off bipolar cells their characteristics. Okay. And then moving back to our ganglion cell layers. We've moved all this information down to our ganglion cells, where they're going to pass down to the optic nerve. So there are 20 types of retinal ganglion cells. Key ones to know are P-type cells, M-type cells, non-M, non-P cells, K cells. And again, just a little point that other people might not clock for exams: ganglion cells projecting to the superior colliculus, the saccadic eye movement. We have slightly talked about MMP cells. So that was something to revise. Moving on. Okay, cool. So let's move on to visual pathways. I haven't discussed, just for the sake of time, colour surrounds, so you can get red on and green off type ganglion cells. But that kind of works through the same mechanism of bipolar cells. So that'd be something I would go look at. But if you understand bipolar cells, you can understand colour occupancy. Cool. Okay, so visual pathway. So we've ended at the retina, and now our ganglion cell axons converge at the optic disc, and this produces our optic nerve. Remember, this is myelinated. So we can carry this now through, and then our optic nerve from each eye will meet at the optic chiasm, where fibers from the nasal retina, so that's the inner region of the retina by the insides of your nose, cross to the contralateral side, and fibers from the temporal retina, so these are the outer bits, remain ipsilateral. So almost, if you think about a pair of glasses, these bits are staying on the sides and the bit from the nasal retina are crossing. So this gives us our optic tract, as we can see here from the optic chiasm onwards. And the optic tract has a few projections, the main one being the lateral geniculate nucleus, the LGN, but it also goes to the hypothalamus, and this is for synchronization of biological rhythms, so circadian rhythms, which is another important role of light. It goes to the pretectum as well, which is important in the pupillary reflex, and it goes to the superior colliculus, which controls the saccadic eye movements by the trochlear and abducens cranial nerves that we've discussed. Cool, okay, still lots to get through. So here is a slide on the LGN. I will just give you 1 second to kind of familiarize yourself, sorry, with that. Okay, I think maybe that was 6 or 7 seconds, but the LGN neurons receive input from the retinal ganglion cells, as I've said, and they're separated into 6 layers, as you can see here, and on the histology image there. And within these 6 layers, we have the processing of the visual information in parallel. So why do we have these layers? Part of the visual organization system. So different LGN layers receive different types of retinal information. So the right LGN receives information about the left hemifield, and the left LGN receives vision information about the left hemifield. This doesn't mean necessarily the left eye, but just the left hemifield of each eye. Okay, and then input from the 2 retina is kept separate within the layers of the LGN. So layers 1, 4, and 6 receive from the contralateral retina, and layers 2, 3, and 5 receive from the ipsilateral retina, as we can see here. And then, as well as being split into contralateral and ipsilateral, the 6 layers are also split into, as we can see, magnocellular and parvocellular, with magnocellular LGN cells in layers 1 and 2. And these receive projections from magnocellular retinal ganglion cells. And these have, again, poor, large receptor fields, good temporal resolution, but poor spatial. Yep, and then our parvocellular NGL LGN cells are in layers 3 to 6, and these receive input from parvocellular ganglion cells. And then these have smaller receptor fields. Cool. Okay. And then again, can't forget, we do have the conocellular LGN cells, and these are ventral to each layer. You can see in the histology there's a tiny purple layer, and they receive the input from the non-M, non-P ganglion cells, and these also will then project to the visual cortex. Cool. Okay, let's move on from there. So now we've gone from the LGN, we move on to the visual cortex, V1, which is the main output from the LGN. And this runs via the optic radiation. As well, you also get output, like we said, from the LGN to the superior folliculus and pretectum. So there's a lot to think about with the visual cortex in terms of organization of information. I think the main things to consider is there's contralateral representation of the visual field. It's V1's organized into a column with each cell layer functioning as a processing unit, and V1 is split into 9 layers and with a distinct cyno-architecture, layer 4 receiving the majority of LGN input, and then this distributes to the other layers. So within layer 4 we have sort of like a checkerboard of receiving from the left and right LGN. So the layer 4 kind of receives everything, processes it, and then splits it into, and then provides this information to the other layers for interpretation there. Okay, so we've gone through some of this, and then, so as I said there's layer 4 with the different blocks for each right and left eye. And then another thing to mention are blobs. So blobs contain monoocular chromatic cells selective for particular wavelengths. They're present in layers 2, 3, 5, and 6. And each blob centers on an ocular dominance column in layer 4C and receives input from KP and M LGN cells, which receiving all input from the LGN cells from layer 4C. And blobs are understood to be involved in color vision, so they're just a further layer of visual analysis. And by, I won't go into it too much now of time, but orientation columns are basically there have been studies that show individual neurons are orientation selective, so they respond best to different to a certain orientation of light shone on them. I think it's really sort of as simple as thinking about that they have receptive fields that are best for certain certain orientations of the light, and they are sort of, these neurons will have quite diverse receptive fields that have been summated from the receptive fields of cells from deep layers and multiple cellular input. And an experimental thing for that is Hubel and Weisel, who showed the different interblob neurons and their different receptive field properties. So that could be something that you go have a look on. I do have a written down version of the study if anyone particularly wants it. It's going quite far. Okay, extrastriate regions. So visual information from V1 is then passed to V2 for further integration and summation. So we have the V5 magnocellular pathway, which is the dorsal stream. You can see that going up, and this extends into the posterior parietal cortex via the middle temporal area. And this is understood to be for motion, location, space sense, control of actions. And then we have the V4 parvocellular pathway, which continues in the ventral pathway and extends into the inferior temporal cortex via V4. And this is for the analysis of form and colour. Okay, that was a brief overview of that. We will now jump on to Audition. just because of time. So again, if you just want to take a minute or so just to gather some thoughts over these images, or if anyone has any questions about vision, please ask now. Cool. Okay, so let's start with the auditory system. So starting at the inner ear, so we have here an image, a diagram, not an image, of the cross section of the cochlea, and this is what is denoted as the hearing organ. So it's a spiral-shaped sort of bony fluid-filled compartment in the ear that spirals for 2.5 turns, and it's longitudinally stretched out divided into 3 fluid-filled compartments. So our outer 2 compartments are called the scala vestibuli and the scala tympani, and these contain perilymph, so they're high sodium, low potassium containing, and our middle media is called our middle compartment is called the scalum media, and this contains endolymph, and that's high potassium, low sodium. Best way to remember this is that media is in the middle, and then just remember the other 2 on the outside. Lovely. Okay, and then as we've got down the bottom here, I've taken this from a lecture, the endocochlear potential of 80 millivolts is across the basilar membrane, and this is because the electrical potential of endolymph is about 80 millivolts more positive than that of perilymph, and this gives us our endocochlear potential, which is obviously important for transmission of electrical signals. Cool. Okay, so off the back of that, we'll now discuss inner ear transduction. So just like touch, or just like physical stimulus in touch, or light in vision, sound and vibration will be the stimulus for hearing, and it'll be our cochlea that transmits this into an electrical signal. So transduction specifically occurs in the organ of Corti, which is on the basilar membrane, which you see there at the bottom. So we have this tympanic membrane, and vibrations of the tympanic membrane are transmitted via ossicles to the oval window of the cochlea. Okay, so we've moved our vibrations from the outside of the ear through the tympanic membrane, recepting these ossicles, then sort of promoting the sound into the oval window of the cochlea. So this transduction, this vibration, then sets up a pressure gradient between the scala vestibuli and scala tympani, which cause the basilar membrane to move up and down. And this is a traveling wave of displacement. And because the basilar membrane is thin and stiff, but it becomes increasingly wider and less stiff, like a fish's tail, I guess, or like a sort of mermaid's tail, it becomes like floppy and wider at the end. This means that this is sort of the first coding point for high and low frequency sounds. So it means that high frequency traveling waves have a maximum amplitude near the oval window, and low frequency traveling waves have a maximum amplitude near the helicotremor. So I think I have another slide on this. Okay, so you can see here within the organ of Corti, on top of the basilar membrane, we have a series of stereocilia, and these are sort of thin projections which lie against the pectoral membrane, and when the basilar membrane vibrates, we get a shearing force between the hair cells, which have the stereocilia, and this gives us a deflection of the stereocilia, and gives us either hyperpolarization or depolarization. So when the basal membrane moves down, our stereocilia move away from something called the kinocilium. And this is the tallest stereocilia that it points towards, and they bend away from this. The tib links slacken and it opens these little at rest shut channels. So it activates these channels by this pulling motion, and this leads to hyperpolarization. However, when the basilar membrane moves up, our stereocilia move in the direction of the kinocilium. Our tip links tighten, the channels open, and then we get depolarization. Sorry, the channels are open at rest. So when the basilar membrane moves down, they close, and vice versa. If you want to do some research on that in particular, the current identity of the neurotransmitter that is then fired when the hair cells become depolarized is currently controversial. So this could be quite an interesting thing to read up on for experimental evidence. Okay, so I've kind of touched on a little bit spectral analysis, which is analysing the frequencies of incoming sounds, and a large part of this is due to the basilar membrane and its changing thickness. Again, a good experimental point for this is von Bexey, and this person was a scientist who did work in cadavers, and yeah, basically found that in high frequencies the peak amplitude of the wave is at the base of the cochlea, and this peak shift moves nearer towards the apex for lower. So that's something that I would sort of recommend dropping in an essay. Does anyone have any questions before I move on? Okay, sorry, I'm just keeping an eye on the time. And then another good clinical point, which I'm sure everyone probably has in their notes already, but furosemide and gentamicin are two antibiotics that cause outer hair cell destruction, and therefore they cause hearing loss. That's something you could think about, the contraindications of providing someone with those antibiotics. Okay, so we're going to move. Ooh, sorry, here we are. So we've moved straight on to auditory pathways. This is a diagram I really like, and I think it's really nice if this is something that you're able to replicate in an essay. I think it just shows a really good understanding of convergence in the auditory system. So we've gone from hair cells in the cochlea, which form efferent fibers to form the auditory nerve. Let me just catch up with where my notes are. And then the auditory nerve, first projection is to the cochlear nucleus in the medulla. Hopefully we can see there. You can see we've got the dorsal cochlear nucleus and the ventral cochlear nucleus. Again, this is something some people might not know, but this is split into two different nuclei, and the cochlear nucleus is important for lateral inhibition, and there's a progressive increase in the amount of inhibition as we move from the ventral to the dorsal cochlear nucleus. Okay, so from the cochlear nucleus, we then project to the superior olivary complex. Just trying to orientate myself. We're going for there. Okay, and the superior olivary complex is will be an important one. It's the first site for binaural convergence. Therefore, it's important for sound localization. So our superior olivary complex again consists of three different nuclei. So this is the medial superior olive, the lateral superior olive, and the medial nucleus of the trapezoid body. So just kind of knowing that extra stuff is always useful. And in for binaural conversions, the medial superior olive receives excitatory input from both sides, so both ears, and the lateral superior olive receives inhibitory input from the contralateral side. So this means that these olives are sensitive to interaural phase differences. And I don't have a particular slide on this, but this is relevant for Jeffrey's coincidence model, if you're familiar with that. As contralateral fibers, obviously, because they're on different sides of the body, it's the nucleus to which they're traveling, have to travel further. It provides a hypothesis for how the MSO is able to detect these interaural differences. We can talk more about that at the end. Required. I'm just going through the main pathways. Okay, and then from the superior olivary complex, fibers then travel in the lateral lemniscus to the inferior colliculus of the tectum, and then from this, information then passes to the primary auditory cortex, which is A1, via the medial geniculate nucleus of the thalamus. No image of this, but A1 is tonotopically organized, so it maps into a columnar organization, and studies have shown that neurons with similar response properties seem to be clustered together into columns. However, the properties of these cells within the same columns appear to be weaker than that of the visual or the somatosensory cortex. They're arranged either in patterns of summation, so this would be binaural information, or suppression, so this would be one ear dominant information, so sort of like if you hear something and you know it's coming from sort of the right hand side of such. Other things for this to pick up is pitch, and this is represented bilaterally in the auditory cortex, and sound source localization is represented contralaterally. Cool, okay. And then the only other things to look at for the auditory system are speech and language, but then that might be a little bit far just for bits. Cool, okay. Taste and smell. For the last time in today's lecture, if you just have a look at the 3 images and have a think about what comes to mind. Okay. Cool, okay. So moving on to taste and smell. Have you got some ideas from that? Let's start by having a look at the olfactory system. Also, I didn't say, sorry, my bullet points have changed into question marks for some reason. They're not questions. Okay, so our olfactory epithelium is within the olfactory mucosa. There's an image of it in the middle there. You can see coming through the ethmoid bone of the cribriform plate. And this is around a 10 centimeter cubed area of mucosa, and it's got 3 main cell types. So olfactory receptors, basal cells, and supporting cells. So basal cells have a stem cell role. So these have regenerative properties, and supporting cells produce mucus. And mucus is actually super important in the olfactory system, because, well, mucus is sort of mainly water with dissolved mucopolysaccharides, antibodies, salts, but odorants and odorant binding proteins can bind to and dissolve odorants to concentrate the odorants in mucus. So yeah, it's actually quite important in the nose and quite an important thing to know. So let's have a look at our olfactory receptors. Have a look at our olfactory receptors, which are right at the top there. So these are true neurons. They're unmyelinated axons with apical thin dendrites, and they form these specialized cilia. You can see this one cilium at the top there, which carry odorant receptors, and they penetrate the cribriform plate there, and they contact the olfactory bulb. You can see that. And these are continually regenerated every 4 to 8 weeks. So again, something topical for this would be the recent studies in COVID-19 patients, and how there's a recent paper in Nature about how they have had, I think, cortical changes in their orbitofrontal cortex, which means they are less responsive to smells, and the olfactory cortex as well as changes in it, which kind of differs from what we understand about the basal cells supporting the regeneration of these neurons, which would be something quite interesting. I wanted to go read on. Cool. Okay. So humans can detect over 400,000 different substances, and I think the main point of understanding the olfactory system is to understand how that is. So each neuron only expresses one receptor gene, so therefore each ORN, each olfactory receptor neuron only expresses one type of receptor, so they're really unique to odors. And once an odor binds to a specific odorant receptor, a sequence of events is initiated that leads to the depolarization of the sensory neuron and leads to an action potential being fired. So basically, an odorant will come along, bind to an odorant binding protein, and then bind to a receptor. And there are loads and loads and loads of different receptors, and they're all slightly different, so they can be triggered by different odorous molecules, and then this gives us a sequence of events that gives us this action potential and therefore codes for the scent. And this is a cyclic AMP dependent pathway. It is actually quite an interesting pathway, so I won't go through the first bit. You know how cyclic AMP is made, and but then this activates, cyclic AMP activates a CNG channel, which leads to calcium influx, and we have high calcium. And then this activates ANO2 channels, which are calcium activated calcium channels. And then we have a calcium efflux, which leads to depolarization. So it's the interesting way of depolarizing neuron. And then this leads to neurotransmitter release. The way we terminate this response is to either unbind the deodorant or excretion of calcium via an calcium transporter. Cool, okay. From that. So you can see we've got all of our little cilia, which would be there. They'd be binding the odorants, and then we move, our signal moves along these branches to the olfactory bulb. The olfactory bulb being the primary target of the olfactory receptor neurons. And it's got a layered structure, which you can see here, which is really nice. So we start with the olfactory neuron layer, which we can see here in blue. And these hold the axons of the ORNs. As you can see here, these green axons that are coming down off the bottom of the ORN. And then we move further down into the glomerular layer, which again is sort of, that's the second half of this blue here, the GL. And this is used for, this is important for information transfer, and we can see here that we're passing on information from the axons to our dendrites of mitral cells and that of periglomerular cells. And the input here is odorant specific. We move further down, we get into the external plexiform layer, and this is just a layer of synapses, you can see. Mitral cell layer contains the cell bodies of mitral cells, and each mitral cell is only activated by one singular glomerulus, as you can see here. So that's really good for maintaining sort of specificity in information flows, as I've described it. Cool, okay. And then we have our granule cell layer, where we have our granule cells, cell bodies, and these again are GABAergic, and just increase the specificity in the system. And then, so you can see here we actually have a really specific system of our olfactory receptor neuron directly contacting glomeruli into the mitral cell layer, and the mitral cells will then project to the olfactory cortex without a thalamic delay, with this inhibitory GABA input from the granule cells and the periglomerular cells. So basically we just have this direct line of input all the way straight to olfactory cortex, with really sort of only two synapses here and then here, and then inhibition from these supporting cells. So really a very, very specific system if you think about how you're able to respond to smells. Cool, okay, and then higher processing. So like I said, mitral cells project to the primary olfactory cortex without a thalamic relay, and they also contact the secondary olfactory cortex, limbic system, and the hypothalamus. So the primary olfactory cortex is used for the emotional, motivational, autonomic, and endocrine responses. The secondary olfactory cortex is used for the conscious discrimination of different smells. Okay. And last of all, we'll just discuss taste. So just to give you a bit of an introduction on taste. So taste, as we know, depends on taste and smell. So I'll cover the responses of sort of taste buds, but also obviously bear in mind that they will be that your taste experience will be influenced by smell. And interestingly, taste perception also depends on cultural and psychological factors, as well as just the chemical and physical properties. Cool, so our stimuli for the taste system are tastants, which bind to taste receptors on taste cells, and this is what provides us with the different types of flavors we experience, so salty, sweet, sour, bitter, or umami. And tastants are non-volatile, hydrophilic, high detection threshold molecules. Okay, and then so we have the tastants binding to our taste receptors. So you can see our taste receptor here with our taste port, again, saliva, just like mucus, really important for dissolving our chemicals. And here's again our taste receptor cell and our basal cell. So again, as these have basal cells, that tells us that they have a really quick turnaround. They have a less than 2-week life cycle. And it means that as these cells are continually turned over, they're all at different stages of development in their life cycle. So you can sort of see at the top here, we've got 8 core microvilli, and these serve as sensory transducers, and these are the receptors for the tastants. And the taste receptor cells basically release ATP or serotonin as a neurotransmitter. If you even think about it now, you probably could taste something in your mouth. So we've got this basal release of neurotransmitter. Cool, okay. And there are multiple models for how taste receptors work, which again, if you have particular interest in this, you might want to go look at. But it's generally considered that taste receptor cells will express one family of taste receptors, and then these are all connected to afferents that are specific for the receptors expressed. So a lot like the olfactory system, specificity in receptors, and then this is summated as we go further on. Really cool. Okay, so not much to know here about central pathways. So gustatory information is carried from the facial nerve. As we can see here, facial nerve, cranial nerve, number 7. And this carries the anterior 2/3 of the tongue, and the glossopharyngeal nerve carries the taste information of the posterior third of the tongue, and these carry the information that's then carried to the nucleus, the solitary tract. Then our secondary neurons pass to the ventral posterior thalamus, and our third order neurons pass the primary taste cortex, which is in the insula, and the secondary taste cortex in the orbitofrontal region. And our orbitofrontal region projects then to the lateral hypothalamus, which is the feeding center, which is why we want to eat more of things when they taste good, or part of it at least. Okay, cool. So we are at 5-2. So that was a very, very, very much whistle-stop tour of 3 important neuronal pathways, or 4 actually. So it would be really great if you could fill in the feedback form, which you can get by just scanning the QR code. And again, you can email me or the preclinical revision email address with any emails. And again, you'll get a copy of the lecture slides if you fill in the feedback form. And if you also want me to go into any more detail with sort of the studies I've been mentioning, or clinical information, I'm happy to do so. Yeah, I hope that wasn't too fast. I know there's a lot more to go into, but I think making sense of the pathways then allows you to kind of make sense of everything else. But yeah, also please just pipe up or put something on the chat if you have any questions.
Hi, any chance you could put the QR, like the link in the chat?
Yes,
I don't have my.
I can do that. I'm gonna type it because actually that's really Do you have any revision questions prepared? I don't have any prepared just because I thought I'd run out of time, but I've got some MCQs we could do. How do I do a poll? I'm not sure how to do a poll. I could do some MCQs. We wanted to do a shout out. If anyone wants to do that, please let me know. Otherwise, I know it can be really awkward to speak up in these things. Or if anyone knows how I can make a poll. Okay.
Would we be able to go through some of the MCQs? Is that okay?
Sorry, go on.
Would we be able to go through some of the MCQs?
Yeah, definitely. Let's do that. Okay. Do you have a preference over what system you'd like?
Probably vision for me,
Yeah.
but maybe whatever anyone else. Yeah,
Cool.
like vision and like hearing.
Cool. Okay, let's do that. So the MCQs I have are okay. I'm gonna write out because if I anyway see my answers if I copy and paste them, which is not a real world, but so. What I'll do is I'll stop sharing and recording, and then